Hello, this is Mr. McMurray again, and we are here at about 10:20, and we're starting on the protein synthesis notes tonight. All right, you won't get to see them until tomorrow, of course, but that's okay. Protein synthesis notes go along with the drawing that we did the other day. All right, so the first step we said is transcription. That's when you go from DNA to messenger RNA. Now, as we said, this is important because your DNA, if something happens to your DNA, then the cell begins to die, and if enough cells die, then you die. So that's not a good thing. So the DNA stays safe in the nucleus. All right, it makes copies of messenger RNA and sends those out to the cell to be used to make the proteins. Now, if something happens to the copies, then the DNA can just make new copies and send them out there. But that way, the DNA is protected, kind of like my originals. I have my originals in a notebook and also have it on a travel drive, thumb drive for the computer. So I don't give you my originals. That way, if you get a worksheet gets eaten by the dog or gets wet or gets spilled on coffee or whatever, I guess lost in a backpack, whatever, then I can just run off another copy from my original and we still get to do our work. How exciting, huh? Isn't it nice to know? All right, so anyhow, that's the same thing with DNA. We keep the original in the nucleus where it is safe. All right, now in order to do this, the DNA has to unzip. So the DNA has to be as chromatin, or the long thread-like wavy stuff, because if it was all coiled up as a chromosome, there wouldn't be enough room for it to open up and for the messenger RNA to see what needs to be copied. Now, so once it unzips, the messenger RNA nucleotides match up with one side of the DNA, because there's only on one side, because they form a single strand of messenger RNA or mRNA. So they only line up on one side, all right? And they only copy the information for one protein or one gene at a time, okay? Now, along the DNA, there may be several different proteins being produced because you're making proteins all the time that need to be used inside your cell. So there may be more than one, but each of those locations on the DNA are just copying one gene. Okay, so one gene is being copied. And that usually means it's anywhere from 300 to 3,000 bases, or the letters, long. This would be like the G, C, A, etc., etc. Okay. Now, for example, if you had a DNA that was C, G, well, let's put an A up here, and a T down here. Okay, if this was the DNA, of course it had the sugars and the phosphate groups over here, but if the RNA are going to come match up with it, well, with the A, it would match up with U, because there is no T in RNA. All right, and there's no I in team, and there's no U in RNA. So instead of AT, you end up with AU. Now, C always pairs up with G, regardless of whether it's DNA or RNA, and that means G always pairs up with C. So those are going to pair up together. And then finally, T would pair up with A. Someone says, but I thought there were no T's. Well, there are T's in DNA, so the DNA strand may have a T, and if it does, then it's going to pair up with an A. Okay, but if there's an A in the DNA, it would pair up with a U, because the RNA cannot have any T's, which it doesn't, and the DNA cannot have any U's, which it doesn't. And so that is kind of how that base pairing would work out. Now, it will do this for 300 to 3,000 bases. So this is a fairly long process, especially for some proteins. All right. Now, the messenger RNA, once it forms, breaks loose and leaves the nucleus through a pore in the nuclear membrane or envelope. Just by the way, these numbers here just represents the numbers of the different things on your drawing. All right. Although this one could be Roman numeral 1 because it's the first part of our notes. But so nuclear envelope is number 15. So it leaves the envelope and goes in the cytoplasm. That is transcription, getting from DNA to messenger RNA. Now we have to do the next step, which is to go from the instructions, copy of the instructions, to the actual finished product. This would be like on Christmas morning, going from the instruction manual with a whole bunch of box of parts and making it into a finished bicycle that works and functions properly. And that's what we're trying to do here. We're trying to take the instructions from messenger RNA and translate it into a finished protein. All right. But the good news is we have messenger RNA. We're going to work with tRNA, and we're going to mess with ribosomes or rRNA. So they're all RNA, so they all kind of speak the same language and easily translate from one to the other. All right. So the information in the messenger RNA is translated into a protein chain. That's what we're going to do. All right, but there's some steps in there. First of all, the messenger RNA has to attach to the ribosome. Okay, and remember, ribosomes are made of R, or ribosomal RNA. So basically, it's another RNA type hooking up with another or different shape RNA type. All right, as you remember, the RNA is shaped kind of like a, whoops, yeah, that's not too bad. Kind of like a mushroom. Okay, this one should be a little curve there. Okay, it's called, we usually just draw them as round when we're doing the cell drawing, but technically there's a little subunit, two little subunits, the big round one and then the little flat one on the bottom. Looks a little bit like a mushroom. All right. Now, the ribosome can read the bases on the messenger RNA and can tell what it wants, okay? But it can't do much about that. It's a factory. Factories don't go walking around looking for supplies. So we need somehow to pick up the supplies, and we are going to use a tRNA here in a minute. Now, we know what we need because every 3 bases on the messenger RNA is called a codon, which is number 7 on our drawing. And a codon is just that. It's a code that tells exactly which amino acid we want. Now, there are about 20 amino acids, and some of them have several different codes that all code for the same one. Some of them only have one 3-letter code that codes for them, so like UGC or CGU or whatever. Okay, and that tells us exactly which amino acid it will code for. All right, now the next step, the tRNA or the transfer RNA, or you can think of truck because it's going to be like a pick 'em up truck. It's going to go out in the cytoplasm and it picks up, because it is a pick 'em up truck, a specific amino acid, and each tRNA is coding for a particular amino acid. It will not just go pick up any of the 20. It will go out and pick out a specific one, and it brings it back to the ribosome. All right. Now, the 3-letter code on the tRNA is across from the 3-letter code on the messenger RNA, which is the codon. So this one is called the anticodon because it lines up across from it, and it sees if it pairs up and matches. If it does match up with the codon, then the tRNA is locked into place and the attached amino acid is hooked onto the growing protein chain. All right, so now we've got the longer protein chain. We only have to do that 99 to 999 times, and we will have our protein. Now, so the ribosome just moves it down to the next 3 bases, or codon, and the process continues, like I said, anywhere from 100 to 1,000 times until the entire protein is built and released from the ribosome. All right. Usually there's a little code at the end that says stop, and it chops it off and says, boom, you're done. This protein is ready to be used, or at least to be to the next step. All right. Now, technically, remember, getting them in the right order is one thing. If they're not in the right order, then you could have a mutation, depending on whether or not it messes up how the protein folds. Okay, but that's the next thing. The protein has to go to the rough ER where it's folded, refolded, and technically refolded 2 more times. It goes up to quaternary, or 4 times folding, all right, to make it the right shape. Very important to proteins that they be the proper shape. If they're going to be a protein in the cell wall, they need to be the correct shape so they can let things in or out depending on what kind of protein they are. If they're a carrier protein, are they a channel protein? Are they a marker protein, the glycoproteins? Whatever. So they need to be the right shape. If they're going to be enzymes, they got to be the right shape, or they will not trigger the right reactions. Okay, so it's very important that they get folded correctly. So this is another place, though, things can go wrong. If they get misfolded, then they will not function properly. All right, so they have to be folded into the proper shape. Shape is a big deal. Now, this protein could be used to make plasma membrane, which is one of the things that the rough ER does. It takes lipids from the smooth ER and proteins, and it can make the cell membranes right there, or plasma membrane. But it could go to the Golgi apparatus, where it needs to have some finishing work done, and it needs to be packaged into a vesicle. And it depends. If it's a hormone or something that needs to be dumped out of the cell, it will go into a secretory vesicle, which means it will be taken to the edge of the cell membrane, and it will spit it out and secrete whatever was in it into your bloodstream. Okay? Or it could be a transport vesicle, and then it's sent off to wherever it needs to go inside the cell, because remember, secretory go to the edge and spit things out transport vesicles just send them around inside the 12, inside the cell, excuse me, not inside the 12. All right, and that is basically it. All right, and it just keeps doing this at a regular basis. Your body is a very good multitasker and is constantly doing these proteins while you're busy doing other things like watching this exciting video or trying to stay awake as you watch this exciting video. And it's doing all this other stuff inside your body nonstop, constantly, without you even thinking about it. So it's a very efficient multitasker that makes very, very few mistakes. Yes, there are some mutations or problems caused when things are not proteins are not synthesized correctly, but for the most part, it happens without a hitch. All right, and that is it for the notes. So we will see you next time, and be sure to stay tuned for upcoming episodes. Good night.